From the first seminar, we learned that complex glycan structures are instrumental in developing diagnostic assays for detecting TB. We now move on to the next seminar, where Dr. Hans Lin focuses on the synthesis of glycans to identify potential inhibitors of a certain glycan-binding protein called galectin. Let me first give an introduction to Dr. Chun-Hung Hans Lin. Okay, so Dr. Hans Lin received his bachelor's degree in chemistry from the National Taiwan University and obtained his PhD in chemistry under the supervision of Professor Chi-Hui Huang at the Scripps Research Institute. He then carried out postdoctoral research with Professor Christopher Walsh at the Harvard Medical School, after which he went home to Taiwan and accepted a position as Assistant Research Fellow at the Institute of Biological Chemistry, Academia Sinica, and rose through the ranks to Research Fellow. Currently, he is the Director of the Department of Academic Affairs and Instrument Service. He also holds adjunct professor positions at the Institute of Biochemical Sciences and the Department of Chemistry, National Taiwan University, and at the Institute of Chemistry, University of the Philippines, Los Baños. Dr. Lin received several prestigious awards for his work, such as the Excellent Young Scientist Award, the Young Affiliate of TWAS from the Academy of Sciences for the Developing World, and the Outstanding Research Award of Ministry of Science and Technology. Research in his lab is focused on chemical biology that involves multidisciplinary areas, including glycobiology, enzymology, synthetic chemistry, and structural biology. Let us all welcome Hans Lin. Hans?
Can everybody hear me? Hello?
Yes, we can hear you.
So I will start my talk. It's also my first time to give online lectures. First of all, I would like to thank the organizing committee to give me the chance to present our recent progress, especially Professor Gladys Completo. So today I'm going to talk about how we spent like several years to develop galectin inhibitors. And before I go to, before I present my research, I would like to thank my collaborators because this work cannot be finished without the enthusiastic effort from my students. I think we have chemistry effort from Dr. Zitu Zijian, IVC, and also the work was contributed by the two talent students, including Crystal and then Dana. So I have to also thank Professor Gladys Completo because so her student Dana Panelas actually spent a year and a half in my lab. Actually, she worked with my student together to develop the chemistry, and also the work also has a lot of structural information contributed by my postdoc, Dr. Lin Xianya. And also I have to thank my collaborators, including Fudong Liu and also Danny Shi, because of their contribute to different expertise. At first, I have to introduce the biological significance of human galectins. So as Gladys mentioned, galectin actually is a galectin binding protein, and they are specific for galactoside, especially beta-galactoside. And so they involve in several biological functions, including cell growth, cell differentiation, inflammation, and the cell proliferations. So therefore, it has been known galectin actually are related to several pathological activity, including the pulmonary fibrosis, neurodegeneration, cardiovascular disease, and so on. And in human, there are 15 different galectins. So they categorize into 3 different groups. including a prototypical, a chimeric, and a tandem repeat. So basically, galectin is a very small molecule. And so the molecule is about 14 kilodalton. And for the prototypical, they are dimers. And then for the tandem repeat, actually, they have two different so-called galectin binding carbohydrate recognition domain. And then they link with the peptide linker. And in addition, and they also can form a chimeric structure for the galectin-3. I think that the challenge in this area is that because there are 15 different types of galectin in human, and they all bind with beta-galactoside. So if you look at the carbohydrate recognition domain, they are highly similar. So therefore, if one galectin is responsible for a disease, so if you would like to develop the inhibitor, so you have to make sure the small molecule, the drug molecule, only specific for your desirable galectin without interfering the function of other galectin members. And so I would like to show this slide. This slide actually mentioned the small company called Electrostatic. They, in 2015, about 5 years ago, they will start with a phase 2 clinical trial in UK. And then the molecule is called TD139. Actually, we will talk about the structural information later on. So this small molecule, they are prescribed for the pulmonary fibrosis for the phase 2 clinical trial. And this is but however, at that time, there is no structural information to understand how the TD139 have a very potent inhibition for the galectin-3. And also, people, they didn't explain if this molecule have a good binding with other galectin molecule. So therefore, today my talk would like to talk, we will develop the chemistry to make a different derivative and then deal with the inhibition potency as well as the selectivity. So this slide actually is an X-ray crystal structure presenting the galectin-3 in complex with the so-called lactose. And so as you can see here, the binding site shown here is a so-called carbohydrate recognition domain, CRD. And then the CRD actually it's not a pocket, actually it's shown as a crevice. They are pretty shallow, and then the CRD can be grouped into 5 different subsite from subsite A to E. So as you can see here, the disaccharide actually is bound with the subsite C and D. And so, as I mentioned to you, the TD139 is a so-called thiodigalactoside. It's a C2 symmetric compound. And TD139, they have the substituent called triazophosphopheniol, and they attach to the 3 position of the galactose. So again, TD139 is also C2 symmetric molecule. So, and as I mentioned to you, at that time, there's no structural information. And then my lab, actually, we present the first structural information by X-ray crystallography. And at the same time, we also synthesize a monosubstituted compound called TSZ-TDG. This is a symmetric compound. So, so here is the structural information for the thiodigalactoside. So if you look at the KD, the dissociation constant, it's about 75 micromolar. And however, the TD139 is very potent, as the KD is about 68 nanomolar. So that is to say, once you attach 2 substituents, so the 2 substituents going to situated at the subsite B and E. And later I will explain the interaction. So the interaction contribute by the substituent is going to to contribute to the binding. And so we at the same time we also perform the so-called titration, isothermal titration calorimetry to measure the binding. So we are able to obtain thermodynamic binding parameter, including the delta G, including the delta H the enthalpy, and also the difference in entropy. And so this way, by this way, we can also calculate the dissociation constant KD. And so this table is quite very complicated, but I would like to tell you is that at that time, we measure the binding for the galectin 1, 3, and 7, and with the 3 molecules, as I mentioned to you, the TDG is the thiodigalactoside, and the TAZ-TDG is a mono-substituted thiodigalactoside, and the TD139 has a 2 substituent. So as you can see here, so if we look at the enthalpy for the binding, so immediately we're going to see once you introduce the first substituent, you are going to have a contribution from the enthalpy. So you will get about 5 kcal per mole. So once you introduce the second substituent, so only galectin-3 has a contribution. And in contrast, when you look at the entropy, again, once you introduce the first substituent and you have a contribution for the galectin 1, 3, but nothing happened, nothing significant happened for the galectin 7. And then for the second substituent, and then you also have the contribution from entropy for the galectin-3. So this is the reason the binding of the TD139 has a significant binding affinity with the galectin-3. And at the same time, we also have the resolved X-ray crystal structure for those 3 galectins, including galectin 1, 3, and 7. So as you can see here, the TD139 is most potent for the galectin-3. The KD is about 68 nanomolar. And then for the galectin 1, the KD is about 220 nanomolar. And for the galectin 7, it's not so potent, so the binding is only 38 micromolar. But if you look at the way how they interact with the galectin, it's quite similar. So I would like to go to the detail to focus on the binding at the subsite B. So here is a superimposition of the complex structure. So the yellow actually is the phenol triazole. So as you can see here, so the yellow is represented TD139 with galectin-3. As you can see here, the fluorine actually can have 3 to 4 so-called fluorine bond with the backbone of the galectin-3. So in contrast, when we look at the complex structure of the TD139 with the galectin 1, because I'm sorry. So for the galectin-3, so the phenyl group actually is has an interaction with the arginine 144 with alanine. So the arginine 144 and the alanine 146 actually function as a clip to hold the aromatic group, so that the fluorine can have a fluorine bond. But in the case of the galectin 1, so the alanine 146 corresponding to valine. So valine is relatively bulkier. So this is the reason the aromatic group actually is tilted 16 degrees. So therefore, the fluorine actually can have less fluorine bond. So they can only have the longer fluorine bond, and then probably only have 1 or 2. And at that time, under such circumstances, they don't have the so-called cation binding, cation pi interaction in galectin-3. In contrast, the corresponding residue is serine 29. So this is the reason. So the galectin-3 has a better binding interaction with the TD139, but not in the galectin 1. So this slide actually shows the comparison between the galectin-3 and the galectin 7. So I already mentioned the interaction occurring in between TD139 and the galectin-3. but if you look at the galectin-7, so the alanine 146 correspond to histidine. So histidine is relatively bulkier. So it's this way. So the aromatic can no longer stay in the subside B. So that is the reason the aromatic has to move away. So this is the reason there is no fluorine bond at all. And then even though the arginine 144 correspond to arginine 31, but because the aromatic already moved out from the subside B, so this is the reason there is no cation pi interaction in galectin-7. So those interactions actually explain the reason why the binding galectin-3 has a strongest binding with TD139, and then less in galectin-1, and then there's no binding at all, no binding interaction at all in galectin-7. So today I'm going to also make more chemistry in thiodigalactoside. So later on we also find out that because we always introduce the fluorophenyl triazole in the molecule, so we can rely on the fluorine 19 NMR to look at the binding signal. So the TAD-TTG actually has only one fluorine, but when we measure the binding in by fluorine 19 NMR, we observe two signals. So this is the reason we discover there's a two binding mode in this TAD-TTG, and so this is the reason we realize that these substituents actually can be can insert into either subside B or E. So that's why we say there are two binding modes for this monosubstituted asymmetric thiodigalactoside. So our idea is if we can convert hydroxy methyl group into the methyl, that is to say, if we can make a methyl group to do the deoxygenation by chemistry, so once the molecule rotates 180 degrees, so because there is no hydroxy group, so that's why because of the loss of the hydroxy group, so they are going to they are going to loss of the two hydrogen bonding. So this is the reason we predict if we can make a deoxy sugar in this disaccharide, so they are going to have one binding mode. And then by this way, we can simply fix the binding. So this is the reason there is no flipping of the molecule in terms of the binding. So at first, as I mentioned to you, we synthesized the two deoxydigalactoside, so-called deoxythiodigalactoside B. It means that they have the substituent at the subside B, and then we also have the other one. The two disaccharides are quite similar, but in this case, we call it deoxythiodigalactoside E. It means this disaccharide has the substituent in the E position, and then the two disaccharides are different. So as you can see here, again, we utilize the fluorine 19 NMR to measure binding. So as we expected, so this one, the TTGB has a binding at minus 114 ppm. So this is relatively upfield. So we assign the signal actually due to the binding at the subside B. And then the other one has a more downfield. And then the binding signal. The binding signal actually is due to the location at subside E. So by this way, so we decide to to make a combinatorial chemistry. So later on I will explain to you chemistry. So if we can generate a number of molecules, so the next challenge is we need to rely on a very robotic screening assay. And then, so the analysis allows us to pick up the lead compound for further optimization. So therefore, we need to have a very efficient synthesis to make a library of a small molecule. And then I need to also explain to you what is the so-called high throughput screening assay. So in the past, we developed the amide forming reaction in the microplate. So it means that we directly make an amide bond forming reaction in the microplate and followed by the screening. And then the group of Polakova, they also using the click chemistry, which is 1,3 dipolar cycle addition to generate many the triazo monocytes and for the substituent activity assay. So here for our thiodigalactoside, we like to use organelle tin chemistry. This means that if you use dimethyl tin chloride, the tin are able to chelate with the cis-diol, and then the less hindered side actually will have a nucleophilic addition to react with benzoyl chloride and to form the ester bond here. And so this is the reason we like to use this chemistry and to install the functionality at the 3 position of our thiodigalactoside. So here is our chemistry. So as I mentioned to you, we have the 2 disaccharide available, including the DE-TDG-B and also DE-TDG-E. So by this way, in one step, we are able to install a different ester group at the 3 position of the galactose. So by this way, we can generate a number of molecules for the further activity assay. And then in this stage, we decide to use fluorescence polarization. It means that our reference compound is also thiodigalactoside, but this compound has a fluorophore. So it means if the compound is in a free form in a solution, so they are free rotated. But once the compound binds with the galectin, so the fluorophore is going to be restricted. So this is the reason they are going to have more polarized light. So they are going to generate the higher signal in the fluorescence polarization. So by this way, by the degree of the polarized light, so we are able to determine the binding level. So I will skip the synthesis. But I would like to point out we start with a very the monosaccharide at low cost. We start with the 1, 2, and the glucopyranoside. The 1, 2, and the 5, 6 group actually is masked by the isopyridine. So I won't go to the detail, but I would like to point out, even though there are many synthetic steps, but most of them, they can be, the synthetic step can be performed without purification of the intermediate product. And then we are able to generate, for example, this intermediate product at the kilogram scale. So as I mentioned to you, we can use organelle tin chemistry to generate ester in the galactose of the thiodigalactoside. And then we quickly generate 116 different derivatives. And then by using fluorescence polarization, we are able to examine the binding with the galectin-1 and 3. So here we are using the level of the binding to generate heat map. So the idea is that we would like to find the molecule which is a very strong binding with the galectin-3, but less with galectin-1. So this is the reason we would like to have selective inhibitor for galectin-3. So here is a molecule, the lead compound generated by the screening assay. So the 6 of them, actually, they are considered to be a good binder for the galectin-3. So, I mean, this table actually summarizes the result. So, the TD139 actually is the molecule which is performed in the phase 2 clinical trial. So, actually, we start with the template, the E, TDG, B, and the E. So, we found out the TD139. We measure the KD. So the ratio of the 2 KD, we define the ratio as selectivity. So TD139, the selectivity is only 3.2. And as you can see here, the lead compound, the B1, E1, and E2, and those compounds, they have the higher selectivity. And the best one has almost 28. It means that the binding for the galectin-3 is 28-fold higher than that of galectin-1. And later on, we also noticed that in the galectin, in the subsite B, there is arginine 144, and then the corresponding residue is serine 29. So our idea is if we can introduce the electrostatic interaction, so if we can generate a negative charge functionality, so we are going to have an electrostatic interaction with the galectin-3 here, but not with galectin-1. So by this way, so we quickly generate again the 6 different negative charge functionality here. So again, we incorporate carboxylic acid with a different alkyl chain lengths. We have 3 carbon, and then 5 carbon, and also 6 carbon. We also generate the benzylic carboxylic acid, but the carboxylic acid, you know, they have it's a meta position and or para or ortho. So and then we look at the binding again. We using we measure the KD by isothermal titration calorimetry, and then use the ratio of the KD to give selectivity. So by this way here, so the compound E5 actually has the highest selectivity, and then the compound E6 has the highest binding with the galectin-3, and then we also resolve the complex structure for the galectin-1 in complex with all those most of the lead compound. As you can see here, we realize that the E6, which is a carboxylic acid at the para to the benzylic here, and then we found out they can have electrostatic interaction with the arginine 144. But however, because galectin-1 only has the serine here, though this is the reason they don't have they either have no interaction or they have a very weak hydrogen bonding. And to understand the high selectivity here is that we found out that this compound E5, and because they don't have any binding, so the aromatic group has to be rotated out of the subsite B. So this is the reason the E5. This explains the E5 why they have a very different binding interaction, and then leading to the highest selectivity. So because of the time limit, so I have to stop the talk here. But the conclusion is that because we can use the organic team chemistry to generate the library of the compound, so it allows us to generate the optimized functionality in the subsite B, and then we found out the negative charge functionality can give a high selectivity and also highest binding. So finally, I have to thank my lab member because of their hard work, and then I also have a special thanks to especially to Dana, and then also Professor gratis completo because she will collaborate with me and then send a very talented student here. So this is the reason we can have a great result for the galectin inhibitor. So finally, I have to thank you for your attention. Hello?
Thank you very much, Hans, for that wonderful talk. So we are now ready to entertain questions from the audience. And Dana Ponelas actually is listening to your talk, and he just wrote a message to you saying, thank you, Dr. Hans.
Yeah, thank you.
Yeah. So Dr. Ruel Nacario and Dr. Gani Padulina, are there questions from the audience?
Thank you, Hans, for that interesting talk. So one question is that, so is it possible for you to extend the structure of your molecule so that you can extend its interaction with the other CRD, the carbohydrate recognition domain A? Because I noticed that it only interacts with domains B, C, D, and E. So is it possible for you to extend your compound?
Yes, I think this is possible. So far, we already have a very good understanding on the subsite B. I think in theory, you can, definitely you can. And then by this way, I think we are able to get more interaction with the galectin-3. Yes, in theory, that's possible. Did I answer your question?
Yes, thank you.
Thank you, Dr. Hans. This is Gani. Hey,
Yes.
Jeff, is there anything on YouTube?
No, there's just like a couple of comments thanking the speaker and also like expressing their amazement. So thank you.
Okay, cool. We have somebody from the audience raising their hand. Mirasol Garcia, would you like to go ahead and ask your question? I wonder if some of them had just pressed press it accidentally, you know. Go ahead. They're still on mute. I don't think we're getting them to talk.
So I have a question, additional question, Hans.
Go ahead.
In your last couple slides, you showed the structures of E5 and E6,
Yes.
right? When comparing its interaction with galectin-1 and galectin-3.
Yes.
So have you tried increasing have you tried placing a linker between your carboxylic acid and the phenyl ring so that you increase the length of your carboxylic acid?
Yes, actually, we made a number of them, and then so far the E5 and E6 are the best. But we are also trying to replacing the carboxylic acid with other functional group, and we are doing a lot of study right now. But so far, the E5 and E6 are the best. But I agree with you. I mean, it's possible to manipulate the chain lengths. And also changing, replace the benzyl with other aromatics. Yeah, it is possible. And then, yeah, that's why we would like to make more variety for testing. Did I answer your question?
Yes, thanks, Hans.
I have a question for Hans. This is Gladys. Again, thank you, Hans, for giving a very informative talk. So my question is actually general. This is also for the benefit of the students who are attending your seminar. So what is the ultimate goal of this research project that we're working on? Are you trying to target a certain disease when you come up with a selective inhibitor of galectin-3?
Yes, actually, there are still several changes in this field. One is actually, they also found that galectin-3 is very important in different type of cancers. So this is the reason they would like to develop the inhibitor. But the problem is so far the best one, TD139, is also very potent for the galectin-1. So this is the reason in this field, people would like to see, more selective inhibitor. So it will be very nice if the molecule can really distinguish between the galectin-1 and galectin-3. And the other change is other galectins are also important in pathological condition. For example, galectin-A and galectin-1. So if we can have a reverse selectivity, let's say if the molecule is more potent for the galectin-1 but not for the galectin-3, I think that will be another breakthrough.
Yes, that's right. So Jeff, other questions from the YouTube live stream?
Yeah, so far there are still no questions. They're fairly quiet over there.
Maybe they're overwhelmed with the structures.
Yeah.
Hi, Dr. Hans. This is Rodel.
Yes.
Yeah, I have a question about these very interesting potential drugs. So do you have any plan, or is there an ongoing in vivo studies on these molecules?
Yes. Actually, we would like to generate a larger library for the testing, because this is our initial try, and definitely we would like to see more higher selectivity. I don't think the selectivity of 54 is satisfying, so that's why we would like to see the higher selectivity, and also we would like to to explore the inhibition for other galectin members. For example, galectin-A and galectin-4, etc. Does it make sense to you?
Yes, and I'm actually interested in seeing how these drugs behave, like probably in mice or rats.
Yeah, actually, you are pointing to the right direction. Yeah, actually, in this field, people also because most of people think galectins only have extracellular function. Those molecules, they only interact with the surface carbohydrate in human and also in the animal. But it's not true. Actually, in the past few years, people start to realize once galectin-3 go inside the cell, they also have the intracellular function. So this is the reason. So now if we develop inhibitors, so we need to explain. We also need to point out how the molecule. So at first we have to explain we are targeting either intracellular or extracellular galectins. I think that's number one. The second, if you target the extracellular, you have to make sure the molecule only stay outside of the cell. But in contrast, if you target intracellular galectin, so you have to make sure that your molecule can quickly go inside the cell and without hanging around outside the cell. That's also another challenge in this field.
Yeah.
That's interesting, Hans.
Thank you.
I have a question. So it internalizes inside the cell. Does it get recycled? Does it go out after? Is it already known?
I think one trick you can do is if you would like to target intracellular galectin, so you can make your molecule very hydrophobic. You can mask, for example, mask the hydroxy group with the ester. Once they go inside the cell, so the ester are going to be removed by non-specific lipases or proteases. And then once they become more polar, so they can no longer go outside. I think that's a trick always used in the pharmaceutical chemistry.
Yes, thank you.
Hello, Hans. There's a question. There's a question from the Zoom chat board.
Yes.
I think this is trying to link it again to the current pandemic, current COVID-19. challenges that we're having. Precious Bernabe is asking, based on this given discussion, is it possible that through your research, can you find a cure or vaccine for this pandemic? Maybe you can relate just in conceptual terms on how.
I think today, what I present here is, I mean, even though we are targeting the galectin inhibitors, but you know, the combinatorial chemistry, the chemistry part, and also the setup of the high throughput screening actually is quite general. I'm pretty sure. I mean, they can be used for other target as well. And because in the past, my lab, we already demonstrate this kind of strategy can be used to target the other enzyme or protein. So I mean, it means those the platform, the technology are general. So certainly can be used for other therapeutic target as well.
What would be your greatest challenge, Hans, if considering bioavailability?
Oh yeah, I think there's a different challenge. I mean, in terms of bioavailability, I think you are talking about pharmacokinetics and pharmacodynamics. So it means that once you have the best molecule, I mean, once you nail down your molecule to 1 or 2, and then you have to examine the cell-based assay and also use animal. Normally normally the mice are used, so you have to use the animal for the study, and then to understand how the molecule can retain in the body, how long does it take, and also you also need to understand if the molecule is modified inside the body, and then also if you look at that, if the molecule can be quickly excreted out of body. I think it's totally different aspects. So we consider. I mean, this is like a animal study, and then you have to examine the parameter in the pharmacokinetics and the pharmacodynamics as well.
Yes. Thanks, Hans.
Yeah, you're welcome.
I have another question, Hans.
Yes.
So, have you considered looking at alternative linker? Because I'm not sure if I remember it correctly. Sulfur-containing compounds, which you have in your structure, right?
Yes.
Have potential toxicity. Have you considered thinking about alternative linker aside from sulfur?
Yeah, certainly. Actually, I think in the past we compare the thiodigalactoside and also the O-link disaccharide, and then so far we understand is the thiodigalactoside are better binder. But I agree with you. I mean, if we have enough manpower, we need to make a methylene link the disaccharide or other heteroatom link the disaccharide, and then to know if because of course the sulfide compound are normally oxidized in vivo and to be converted to sulfone or sulfoxide. So I mean, of course, we have to consider once they are oxidized, does the resulting molecule still retaining the activity? And then the other issue is, of course, we need to look for other linker, and then to know if they are also good binder. Because if you, for example, if you have the methylene linked disaccharide, definitely they are more stable, and then it will be nice if they still retain the same degree of binding as well.
Thank you, Hans.
So are there more questions? I think 2 participants raised their hands.
Oh yeah, there's a question here in the Q&A portion from Michael Alias. So the similar question as what actually similar question on what I asked earlier about sulfoxide or sulfones as linker. Yeah, it's already been answered. Let me see.
Two participants raised their hands, so maybe they have a question. I just don't see who are these people.
Hi, Dr. Hans. This is Dana.
Oh, hi.
Hi, how are you?
I'm fine in Los Baños. Dr. Hans, I would like to know if you mentioned earlier that you want to generate a larger library
Yes.
or to come up with a more selective and more selective inhibitors. May I ask if do you plan to use thin chemistry also to introduce different more functional groups using the ether linkage? Is the chemistry more challenging than?
Okay, let me explain. Let me use two examples to explain our current problem. Is one is the thin chemistry. We found out that in some of the reactions are not high yielding. So this is the reason they will generate variation in the library. So probably in some well, you see the very good yield, and then the amount are much more. But in some wells, the yield are not so good. So I think they are creating some variation in the result. So that's something we have to overcome. We need to find a better catalyst. That's number one. And second is that we found out that in some of the screening assay, the ester are not so stable, because the ester are supposed to be, stable if the pH is below 8. But if your condition are more basic, the ester are quite labile once the pH is above 8.0, even 8.5. So this is something we need to avoid. So that would be nice if we can, install different functionality like the ether type or other functional group which are more stable than ester. So that will be, provide more reliable result in the screening assay.
Thank you, Dr. Hans.
You are welcome.
So I think that's it. Thank you very much again, Hans, for giving this very informative talk. And for those students—
thank you very much, Gladys. You did a lot of work, and thank you for the success. Thank you.
Thank you very much. So for those students who are watching, if you're interested in Dr. Hans Lin's research, feel free to contact him, and you can see, you can either contact us if you want his email address, and it's also available in their website, right, Hans, if they want to contact you?
Yes, no problem. Yes.
Yes. Again, thank you very much. And I hope you can still stay if you don't have meetings after this, and you can listen to the other seminars. But
yeah,
yeah, thank you so much.